the button and I started it. And now I have to see if I can actually get things to play because
Why does
apparently
my rundown
iTunes is
keep disappearing?
updating and can't work while it's updating. Although I never told it to update. So what is going on here?
All right. It's okay. I need a second anyway because my
Technology.
my
Wait, hold on. Can I get this to
I
work?
keep
Is the
closing it.
question.
Oh, I see what it is.
I can get it to work there.
My rundown is
It's
doing
good.
loady circly thing.
You're doing loady circly. I got music.
What is going on?
We got music. I just have to make sure that I can play the music. We got all the music that we need now. I have all the songs. I'm not. No. You know, I do not want to install my updates right now. I'm kind of busy.
Yeah, my computer just said that too. I was like, try tonight.
How about remind me tomorrow? That'll be later. That'll be later. Hey, everybody. We're recording, but we're not recording. Justin, are you ready to go?
No.
You still have spinny wheel of death?
I don't know what's going on. Yeah, I have all of a sudden I've got this all over my interwebs. And okay, things are starting to settle down.
Your internet. Okay. Your internet's.
I might have choppy internets tonight. Choppy CZ. We'll see.
Yes. So I'm going to announce for everyone. I apologize. We were going to try and get a scientist from Fermi Lab on tonight to talk about dark energy, but it's not going to work out for this week. But we will have a scientist on February 6th. So it's on the calendar, February 6th. We are making that happen. And so you can look forward to a dark energy conversation at the beginning of next month. Are you ready to start this?
Born ready.
Born ready. All right, let's count it down. Starting the show in 3, 2. This is TWIS. This Week in Science, episode number 705, recorded on Wednesday, January 23rd, 2019. Driving and cloning. As you do. Hey, everybody, I'm Dr. Kiki. Tonight on This Week in Science, we are going to fill your head with sexy smarts, cures, and clones. But first...
Disclaimer, disclaimer, disclaimer. The wonderful thing about wondering about things is the wonderful places that wondering can take you. No matter how far back you go in current human history, there's evidence of wondering about the unknown. And one of the wonderful places that wonder often leads is discovery. Discovery can be the destination of wonder, but it can also be the journey itself, with many discoveries whooshing by as you go. And along that wandering journey of wondering, the many discoveries unwound from the unknown will undoubtedly wheel us towards more wondering, until one wonderful day when we discover that what we've all been wanting all along is simply another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
Good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the science that we loved from the last week. Stories we thought were amazing and worth sharing with you, our favorite science friends. Yeah. So on this week's show, I have stories about clones, about gene drive, and maybe some cures. We'll see about that. But Justin, what do you have?
I've got a cure for cancer. Maybe.
Oh, for mice,
Maybe.
right?
Maybe. Well, that's maybe. That's always the thing with the cures for cancer on the show. It's always maybe. Also, I have, of course, some ancient human, even some Neanderthal news, as well as be careful what you say because the flowers may be listening.
Uh-oh.
There are snoops. Snoops in my yard. Snoops in my kitchen. Blair, what is in the animal corner?
Oh my
Listening
goodness.
animals?
I have 2 cases of very smart birds, and
Cases, is that like 24?
Yeah. Yes. A bushel of smart
bushel.
birds.
Right.
And I also have a particular case of a lizard miracle.
Wow. All right. I am ready for some miracles. We need some miracle. Everyone, as we get into the show, I would love to remind you that you can subscribe to This Week in Science as the podcast that it is, all places that podcasts are found. Look for This Week in Science, or you can go to TWIS.org to find information about how you can subscribe on iTunes, Google, YouTube, and then there are other places like Stitcher, Spreaker, TuneIn, Spotify, Pandora, all the fun places. Find us because you don't want to miss stuff like this brand new paper that came out from Mike Brown and Antonin Batkian, who I'm not pronouncing his name, Konstantin Batkian. I'm not pronouncing his name right, I'm sure. 2 Caltech authors who have been looking for evidence of the existence of Planet 9. Number 9. So, Dr. Brown came out on Twitter this week with a nice little thread that he said 3 years ago, he and his co-author, Konstantin Batygin, maybe? I'm really not helping here at all. And I proposed the existence of Planet 9.
Every time you'll probably be right eventually.
I'm trying. I'm going for
right
your
one.
scattershot method. Exactly.
Yeah, that's how I do it.
Anyway, they proposed the existence of Planet 9 because they noticed 2 strange things about the most distant objects in the solar system. First, Kuiper Belt objects with distant, very eccentric orbits are predominantly aligned in one direction. And that, I mean, unless there's something acting on them, they should be fairly randomly oriented. And secondly, the orbits of those Kuiper Belt objects are mainly tilted in one direction as well, which is also kind of strange, right? So they proposed an idea of a distant, massive planet on an inclined, eccentric orbit. So not on the exact plane of the solar system, as it were, but on its own little path. And this provides an explanation to this. And so that's where their hypothesis came from. They have now published an article that has come out this week. You can probably find it on the archive.org. I think it was supposed to be available as of today, but the paper itself is published in, where was it published? I forget. There's a paywall for the actual paper where it is officially published. But you should be able to find it, not with a paywall, at the archive. Anyhow, this new paper looks at actual data from these Kuiper Belt objects, asking the question, are they aligned? And so they have a bunch of the orbits of these Kuiper Belt objects, and they find that there is really alignment, and it doesn't seem to be very random. And the average of all the data ends up in one very specific place. Moving on from there, he's like, well, let's look at this, and maybe it's a random fluke. And so they simulated 100,000 observations of non-clustered, so Kuiper Belt objects that are not all clustered together, objects, calculated the average of those, and from those actual observations of objects and the data from those objects, determined that this is this is pretty different from where what they found with the clustered objects. So these clustered Kuiper Belt objects are very likely being influenced. They're all kind of clustered together. They did another analysis looking at the tilting, and they discovered that the percentage of error is very slim. And so by the orbits and the tilting and running a multivariate, a multifactorial analysis of the variance on these things, they calculated that they had basically a 1 in 500 chance that what they found is a statistical fluke, which is pretty good, right? And so there was other data that potentially discredits, or it doesn't corroborate, not necessarily discredit, but doesn't corroborate what they have seen in this Kuiper Belt object data. But they went and looked at that data specifically and determined that they weren't really able to be looking at what they were looking at. Because, so from the paper, they say, because of the limited survey region and small number of detected distant objects, no conclusions on clustering of longitude of perihelion observed in the complete dataset can be drawn from the OSOS data. And so they think that's the most complete analysis that can be done on the data, this OSOS data set, that would not necessarily corroborate what they found. And basically that data set can't draw any conclusions because it's not complete enough. They need more numbers of clustering objects to be able to make any conclusions. So, okay, the journal article in the Astronomical Journal, that's where it is. But the basic take-home message from this: the cluster that they have found is real. The tilted orbit the tilt to that cluster is real. And it could potentially be a cluster of objects. And it's hard to tell the difference between one massive object and a cluster of objects, very closely spaced. And there is another paper out this week that was talked about in the media suggesting that, hey, maybe it's just a big cluster of objects and not a planet. Which doesn't actually look at any observational data at all. And so Mike Brown has come back and said, well, they did modeling, which is good, but we actually looked at observational data. We found data that looks like there is something there.
That's really interesting.
But I mean, I kind of get the point of the clustered objects, because at a distance, their gravitational effect would be the same as one solitary object would be. So I kind of get that, but yeah, but this I mean, this is a very intriguing question, that we could have this other planet in this really wildly misaligned orbit, probably a retrograde orbit, I would assume, to help explain why we haven't captured it already in some telescope somewhere for a prolonged period. But let's get more data.
So let's chase it. I mean, they say, they really think, well, at their last sentence of their abstract is, while explanations other than Planet 9 may someday be found, the statistical significance of this clustering is now difficult to discount. Dun, dun, dun. What is it out there?
It'd also be such a bummer if it passed by close enough to have reached, but we weren't prepared to go, and we won't see it again. And
no, it
is out. We could send a thing, and now it's gone, and it's
gone. We won't see it again. Another 100,000 year orbit. No. But going away from these far off things that maybe, lead you to think, how about thinking about some things here on this planet? I would like to start a story with a segment of the show called This Week in, oh my God.
What? Oh,
what are we doing? No, the science is sound, but there are definite conversations that are going to come from this story. Chinese researchers who successfully cloned 2 macaque monkey babies last year. We're not talking about He Jiankui's human CRISPR gene edited trial. What I'm talking about, they cloned 2 baby monkeys. That was the first time a primate has been cloned. Well, they
Right.
decided they were going to take these things and let's, put these worlds together. And so they've now published 2 papers in which the first is the production of a the production of a CRISPR edited embryo. And so they edited an embryo, a macaque monkey embryo, using CRISPR-Cas9 to take care of a gene that's called the BMAL gene, BMAL1. This gene is very important in our circadian cycles. So taking out this gene basically just destroys circadian regulation of many
Danger.
So you have a monkey that can't sleep, basically.
Not that, but cellular. I mean, if you're talking cellular circadian rhythms, you're talking about the health and function and behavior of a cell, how it grows,
healing, memory storage,
the whole.
Yeah.
So they created gene-edited donor monkeys without the BMAL1 gene. They exhibited circadian disorder phenotypes. They had reduced sleep time, elevated nighttime locomotive activities, dampened circadian cycling of blood hormones, increased anxiety and depression, as well as schizophrenia-like behaviors. And it's expected that these kinds of monkeys, they would be very useful as being an edited organism to study circadian-based and potentially neurodegenerative diseases. We could use monkeys like this.
Or if you wanted an army of schizophrenic monkeys to unleash upon the world.
Well, that was their next step. I mean, they're not unleashing an army upon the world, but the next step is that they published a second article taking donor cells, fibroblasts, which are effectively skin cells or connective tissue cells, from these gene-edited monkeys, inserted them used stem cell techniques and somatic nuclear transfer, which is the technique used to create Dolly the sheep, the first clone. And these edited cells, the donor cells, were inserted into the eggs that had the nucleus removed. So offspring were effectively clones of the donor. And they looked for the best representative of this BMAL1 editing phenotype and genotype that they were hoping to get. And they say that this line of research will help to reduce the amount of macaque monkeys currently used in biomedical research around the world, without which is great. Yes, without the interference of genetic background. A much smaller number of cloned monkeys carrying disease phenotypes may be sufficient for preclinical tests of the efficacy of therapeutics. That said, they are planning to improve the technique and increase the number of cloned macaque monkeys that they produce in the future.
What is the benefit of using cloned macaque monkeys instead of breeding the monkeys that were CRISPR'd?
So my first guess would be just what I was reacting to there. What Kiki said was that the number of, and this is air quotes for those of you who are only listening, throwaway monkeys.
Right.
The carnage that comes in animal research, which nobody working in the industry is benefited by. Is because of you're looking for a very specific subject to do your experiment with. And if you don't have it, it has suddenly no value upon the earth and cannot be released into the wild.
Well,
the more narrowly you could make those selections, the less of a breeding program you would need. And so the less, to put it in no other terms, carnage that would ensue.
So just a little bit of devil's advocate here. What I know about primate research, which again is not a huge amount, but it's some, is that usually there are primate centers that are used for research and that there are different subsets of monkeys there used for different research projects.
Okay.
So the likelihood that you would need 100% of monkeys at a research facility to have the same genetic expression for all of the research being done in that space is low.
No. It's saying for instead of going through a breeding program that could reach into the thousands to achieve a subset of subjects that you could then send to that primate research center, you could literally make the 5 or 6 that they need in a lab, as opposed to doing it.
And
I mean,
and
think of the Jackson Lab folks.
Yeah, that's what I was going to say.
Yeah, right?
That's what I was going to say. So instead of mice, which we know if things work in mice, there's a rare how long is it going to take to get from mice to people? There is a long pathway for drug transfer, for treatments to actually get down the pipeline, because there are huge differences between mice and primates even, and people. And so if we can do some of this research on primates and have primates that have been genetically modified as mice have been genetically modified, rats have been genetically modified by the Jackson Lab and other places, it could, as has been said, reduce the number of animals that are needed, because you're not fighting against random variation of genotype.
Yeah, and also I overshot your question a little bit, Blair. My comment about the Jackson Research Lab was my amazement that they could CRISPR edit the mother and the father and still have offspring that had the other genes in them, which kind of blew my mind. That it wasn't 100% heritable, because of course epigenetics, it could be the grandparents, the great grandparents seeping in, and so even in the CRISPR model of producing what it is that you want, there's a high enough rate of error, or it not being showing up what you want in that next generation, that the cloning would be a better, more efficient, possibly. And according to this, possible. This is a way to go.
One other question before we move on. How do you know that the CRISPR process is not affecting other things in the monkey? Because sometimes there's multiple factors in a locus on a gene. And so I feel like there's definitely, and maybe that's what's next, but there's definitely some testing to be done to make sure when you kind of engineer a particular test group that you're not accidentally creating confounding variables.
Yeah. So with this kind of situation where they're creating the gene edited animals first and then the clones from the gene edited animals, they're looking at the genome, the genotype of the ones that have been edited, to look for the ones that have the highest accuracy. So their genotype, they will go through and genotype all of those organisms, all of those animals. And so, for example, out of 325 somatic nuclear transfer embryos, they had a whole bunch. There were 65 surrogate monkeys. So let's say for the knockouts, let's see if I can find the numbers there. I don't have the numbers there, but
this is also sort of a temporary question. There's next generation. I don't know how many generations in we are. CRISPR technology that is going to have a massively greater efficacy, no off targets or very few, and also be able to be applied to a much broader spectrum of the genome than it is currently. So that those questions about how effective or the off targeting of CRISPR are just sort of limited to this time now in which we speak about it, and will sort of just go away naturally as the technology progresses.
Well, I mean, I understand that the technology itself will get better and more precise, but I more mean that there's gene linked traits that we don't know about in every genome.
Right.
And so you could remove something that you want to remove and inadvertently affect the animal in another way.
Yes, but then you might discover that. I mean, that also might, just the fact that you didn't get the question you asked answered, but something else was answered, that's still getting the job done.
And that's something that has happened for a long time in gene knockout animals.
Oh yeah.
This knocking genes out or putting genes in, knock-ins and knockouts, this has been happening for a long time without CRISPR. Researchers have been doing this for a couple of
And
decades
it's been
now.
happening through evolution.
And it's been happening,
That's
but-
all of the things that we're trying to fix.
But at the same, but in a research sense, when you're trying to, like you said, you knock something out and something else happens, potentially your question that you were asking isn't answered, but you learn something new. And so that is part of this. That absolutely is part of this. Yeah, there's a lot going on here. There are people, I'm sure there are many people in the audience who don't want to see animal research like this take place. But animal research at this point is still a necessity. And if we can reduce the number of animals that we are using, that is a good use of science.
And also, I can tell you that I've never met
Absolutely.
a researcher who works with animals who said, I wish this was less efficient.
I do.
No,
I do. What I do hope, though, yeah, everyone wants it to be more efficient.
Yeah.
But what I do hope is that as we're pushing this, bleeding edge of technology and scientific and advancement, I really hope that all the countries that are and institutions that are invested in it are playing by the rules that we have set for animal research, because
Right.
human research,
going
animal
to-
research, there are standards, and
Yeah. Well, we
They're
know that's
important.
not true. We know that's not true in the way
I
that
can hope.
different countries treat humans. We know this is not going to
Yeah.
be true. So it's a great hope. Poof,
Yep.
there it went. Okay.
Oh.
All right. Well,
right.
moving on. If anyone wants to talk about it, talk to us about it on Twitter or send us an email. Let us know. But this is This Week in Science. Justin, tell me a story.
I'm going to tell you a story, and it's not the one that's in the queue, because I got sent a story moments before going on air that blew my mind like no story has done in a very long time. Okay. So it's sort of a pop quiz kind of thing. I want you to design something to catch or amplify sound. And I'm not talking about electronic software or anything with an analog, like a physical thing that can catch or amplify sound. What would that device look like?
Like a cone, like a
Like a cone,
yeah.
A parabola.
funnel kind of a
Dish,
thing.
yeah.
Something
An
that
ear
would take a
trumpet.
walk-
What's that
a big ear
do?
trumpet, right.
Yeah.
And the design that took you no time to come up with is so fundamental to acoustic collection that it might not be so surprising that something evolved this shape over billions of years would have the ability to hear, like different shapes of ears,
Mhm.
right? And if I was talking about different shapes of ears, focused on picking up different frequencies of some sort, or different wavelengths of sound, or different types of sound, this would seem like I was telling you an obvious tale. But if instead I told you I was talking about the shapes of flowers and their ability to pick up acoustics from the environment, that's when you go, what?
So
Enter,
a long time ago, I did a story about bats using blades of grass that were tubed as projection to be able to echolocate. Yeah.
So this is where we have Tel Aviv University researcher Lilac Haddany, whose name I might totally be mispronouncing. Haddany's team looked at evening primrose and found that within minutes of sensing vibrations from pollinator wings, the plants temporarily increased the concentration of sugar in their flowers' nectar. In the effect, the flowers themselves basically worked like you said, like the ear horn, right? So they were able to pick up very specific frequencies of, in this case, bees' wings, while turning out and sort of filtering out sounds like wind, rustling, other plants who were probably also trying to attune themselves to the passing bee. To test the primroses in the lab, Haddany's team exposed plants to 5 sound treatments: silence, recorded sounds of honeybees from 4 inches away, computer-generated sounds in low, intermediate, and high frequencies. Plants given the silent treatment placed under vibration blocking glass jars had no significant increase in nectar sugar concentration. Same went for plants exposed to high frequency and intermediate frequency. But for plants that were exposed to the playback of the bee wing sound, and also similarly in the similar kilohertz point, 0.2, 0.5 to 0.5 to 1 kilohertz, the final analysis revealed an unmistakable response within 3 minutes of exposure to these recordings of the bees. So we're not talking about molecular exchange in the air or other ways that we know that plants have been able to communicate in the past. But a recording of these frequencies, sugar concentration in the plants increased from between 12 and 17 to as much as 20%.
That's a lot.
Within 3 minutes, too. That's so fast.
And right away, yeah. So I mean, because this is one of those things, okay, like, okay, wow. Now, if we had this knowledge, you look back at it, and production of nectar is such an expense for a plant, right? Is that the biological expense of producing, that the efficiency of being able to produce it when you really could benefit from it makes more sense. To benefit, to create it in a burst that will benefit you directly without having a sustained burst the whole time, all the time, every time. Huge evolutionary advantage in being able to do that. And then so I mean, and we think about, it seems really weird that flowers would have ears, but we know that leaves, sense light. Like
also
eye senses light.
They also sense odors as well. Plants,
molecular odors. They translate
react.
that, as we talked about, right?
And we've talked about how the roots of plants emit a clicking sound when there's danger.
Yeah.
So like
very attuned to these vibrations.
Yeah.
And really, animals are just organisms that respond to their environment, right? In an active way. And we can just think of plants as animals that don't move.
and how much more important that
they're just animals that can't run away.
How much more important that they have all of the ability to sense what's going on around them, and the fact that they can't. You think you need senses when you can just leave.
Yeah, right? For real.
I'm getting out of here. Well, no. What if you have to figure out how to make do with what's around you? What comes to you? What's going to attack you where you are?
And I'm having this funny debate in the chat room with Dave Shorty about what the difference between responding to a frequency of sound and listening to something is, because it's pretty. Is there a difference between responding to a sound trigger and having an ear?
Yeah.
I would say behaviorally. I mean, it depends on what level you're looking at. Are you talking about a neurological response? Or are you talking about a behavioral response? Because when I think response, I think a physical action, even if it is blinking because you heard something. That's still a behavioral response, even though it's not a big one.
Well, and we might have a big clump of nerves that takes those things that we hear, those frequencies, and turns them into things that become more of this kind of big deal. But it starts in the same place, which is receiving these frequencies.
Yeah, it starts in reception and response, and a positive response or positive reaction and response. It benefits, and this is evolution. Flowers have been doing this for a billion years or more. I mean, more. And I think
I looking at this this little conversation that's taking place in our chat room. I think what we're getting at here is whether or not there's comprehension, right? And it doesn't necessarily matter. I mean, at what level does comprehension occur? Are we talking about consciousness and understanding of that's a bee? I need to, knowing it and saying, I need to increase my sugar output. But, we have lots of, unconscious reactions to things that we don't realize. You know, our pupils dilate. But, you know, we flush. Our skin flushes with blood to release heat. There are all sorts of things. Our hearts start pitter-pattering faster when we get excited. And these are things that are from sights and smells and sounds that we are similarly tuned to. And so comprehension is its own, is a different thing. And there are some, and I think it's a spectrum. I would say it is all a continuum.
So what's also interesting, like in the cognitive sense, I would say that this isn't a well-planned response from the plants in a way.
Planned.
Right. In that or I mean, it's what is
it's more of a reflex, right? Like he was saying,
it's like efficient way to do it, but it works, right? So one of the things that the story points out is that even though they're doing this 20% increase to really bring in the crowd of bees, bees themselves actually are susceptible to noticing a 3% increase, sometimes even less.
So the plants are like overdoing it a little bit.
Overkill. Big
They're
time.
like, I want to be your favorite flower. Come back to me.
Yeah. So right. So maybe that's even okay. Then I got to take back what I said about this not being necessarily the most efficient. Maybe they're also competing against other flowers.
That's
maybe
exactly what they're doing.
Studying other flowers, you find that some only come up with an 8% increase. And so the 20% is going to, and then there's also, they also are like, now like, okay, now we got to look at what flowers are attracting what pollinators. And is there a variation in their ability to pick up the acoustics of the different wings and the different frequencies put out by the flying insect in comparison to the shape of the flower? So there's this whole now. Oh my gosh. Anyway,
there is a world to explore there. Oh, evolution, you're so grand. It's this kind of a story that just makes me just full of awe at the universe and our planet and the life that is on it. It's amazing. Is it time for us to jump to Blair's Animal Corner?
Yeah, let's do it.
Oh, don't open iTunes. Dead side. Oh, it's gonna work. Okay, great.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. Hey, man, I'm
what you got, Blair?
Did you know that crows are smart?
Of course I know. I've been listening to you on this show.
Oh, yeah, okay.
And didn't we have
time telling me how smart crows are?
Yeah.
And didn't we have Kaylee on here a while back?
We sure did.
Orbit expert, yes.
I just
when I went on vacation, didn't a crow sit in for me and actually do my part of the show?
That's right.
I just have the latest headline in the series of Crows Are Smart stories. This is, of course, the New Caledonian crow, the superstar of crow intelligence. This is a study from the University of Auckland, the University of Cambridge, Bertha von Suttner University, and the Max Planck Institute for the Science of Human History. Interesting addition there at the end. They found that New Caledonian crows can infer the weight of an object by watching how it behaves in the wind. So we know by looking at things how heavy or light they are by the way they respond in breezy conditions. The study they offer up, or the example they offer up here, is that if you have a napkin and a fork on the table at an outdoor cafe, the napkin will fly away, the fork will not. You might even take that fork and put it on top of the napkin to weigh it down. But we have not studied any other creature being able to figure this out just by looking yet. This might just be a case of not looking. I haven't heard a lot of studies looking at this specifically. But regardless, this is a study where they didn't use lab birds. They went out into the wild and grabbed 12 birds from the wild, brought them back into the lab, and they were taught to use a weight of an object to receive a food reward. So half of them were taught that they needed the lighter object to get their food reward, and half of them were taught they needed the heavier one to get their food reward. Then they took those same objects that they learned about, they strung them a few inches off the ground, one by one, pointed a fan at them. They got to see how they blew around. And so even though they might look the same, by watching which ones moved and which ones didn't, the ones that blew around easily, in theory, were lighter. The heavy ones remained stationary. Then they brought the birds into the test area, and the birds got to pick whichever one they wanted without having touched them first. So they picked something and it was over. They didn't get to pick them each up and decide. And they were 73% accurate. So definitely more than just random in choosing the object that would get them the reward. So if they were taught that they needed the light object, they went for the one that was moved around by the wind. If they were taught they needed the heavy object, they picked the one that didn't move from the wind. So this is a very interesting bit of kind of inference that they are able to figure out just by watching natural conditions. Also, they tried to do something that they might actually see in the wild. Wind is a very natural thing. They grabbed these wild birds, so they're not used to solving puzzles.
Right. These aren't their lab crows that are like, hey, have you done the latest New York Times crossword?
Yeah. Oh, I think they call this a rock. Back in
I solved Rubik's Cube in 7 seconds.
Oh, yeah. So these wild New Caledonian crows could figure out by watching how things moved in the wind what was heavier.
I wonder if a wild bird would have an easier time than a lab-reared bird.
Oh, they don't know from wind.
They don't know wind from, right.
Having
indoor crows.
Wind. Yeah.
That is a good question.
Yeah. And what other bit of physics does a wild crow come up, or any wild bird come up against all the time, but the effects of wind in the physical world? I actually was watching, it wasn't a crow, it was some small bird on a windy day not too long ago, as the branch was oscillating up and down. And the bird was I was actually both oddly curious as to why it didn't just take off.
Was he adjusting so that his head didn't move at all?
So the bird was both showing me this amazing physical trait of being able to balance on this wildly moving thin limb, because there's big limbs it could have moved over to, not really moving at all, but it stayed on this one really big moving. And then also why not change? Why not move? And it seemed to it'd be fine. I can handle this. I can totally adjust my weight.
It was like a bird teeter-totter that didn't require a partner.
Yeah, maybe.
See, if birds could go wee, you might have heard that, but they can't. Maybe New Caledonian crows can. We'll find out, perhaps.
Yeah, and then the other question is: we know now these New Caledonian crows have amazing mental capacities. What other corvid cousins can do this? How far does this ability go? Because the puzzling ability, their ability to solve the puzzles, doesn't appear to doesn't go that far in the corvid world. We don't see other species really able to pick up these puzzles the way that they do. But something like telling how much an object weighs, I think that's going to be a very basic understanding.
Yeah, I think if we could find a way to test it in a way an animal would want to solve it. For some reason, I have this impulse to think that a lot of animals could do this, because the wind is such a universal, and weights of things are really important for if you're digging something. If you're going to put something in a hole, you want to weigh it down. If you're trying to pick the nut that has more meat in it, you're going to pick the one that's heavier. I feel like there's some universals here that we might see in other taxonomy potentially. Absolutely. But we'd have to figure out a way to test them. Crows are extremely easy to test in these sorts of things because we know how to teach them things and we know how to incentivize them.
Yeah, but wait. Don't tell me.
How does it transition to your next story?
Oh, wait, don't tell me. Oh,
Instead of
Which I can't
wait,
see from here,
wait.
but I'm just assuming that's right.
thanks, Justin, for
It
the
was an awkward
assist.
3-second
But
pause.
birds are really smart. Is that right? Okay, yeah. So the fact that birds are so smart might actually be evolutionarily advantageous to them. There's actually a Darwin theory that mate choice could contribute to higher intelligence in some species. And a new piece of research from the Chinese Academy of Sciences in Beijing and Leiden University in the Netherlands looked at, I love how they say this in the article, 34 small Australian parrots known as budgerigars, aka parakeets, aka budgies.
Budgies.
Budgies, beep, beep, beep. To test the notion that a suitor's smarts could outweigh style or songs. So they wanted to see where intelligence factored in mate choice for budgies. They took a female bird, they exposed this bird to 2 similar looking males. She could only interact with one at a time. So she had to pick A or B. Bachelor number 1, bachelor number 2. And so prior study designs like this showed that females would pick the male with the nicer appearances or better songs. One or the other. It was always appearance or songs that won out in those cases. They could tell, obviously, which one she preferred by how much time she spent with each of them. And then, they took the lesser male, the loser. They took him and they gave him a special training session. And they taught him how to open a container filled with seeds that was difficult to open.
Oh, I like where this is going.
Yes. So then the female and the winning male, the macho guy, received no such training. And then they were given open boxes of seeds to just eat from freely. They lived the life. But then the female was put back in the cage with a sealed box of seed. Mm-hmm. And she watched the trained male open his box of seed. And then she watched the untrained male, who she preferred before, unable to get to the seed. 8 out of 9 females changed their mind, spent their time with the male who could open that box. So the researchers posit that this supports their hypothesis that started with Darwin, that sexual selection could affect the evolution of cognitive traits or intelligence across animal species. Showing intelligence could help somebody get a mate and better spread the DNA. Now, here is the big question mark here.
So, can I say one thing before?
Yeah, go ahead.
There seems to be a little bias in this study that is being done by educated people who may not themselves have always felt like the most physically attractive.
Yes, they want to be told that they have the evolutionarily advantageous trait in this case. Yeah.
See, it's actually intelligent. As much as I would like to believe you could get by on wits alone, there's another element to this, which is only one of these birds had seeds that she could eat.
Yes.
Like, aside from how they got them, it meant nothing.
You have food.
So this is exactly
dinner,
what
nobody else is.
George Strider and Nancy Burley of University of California, Irvine, gave an accompanying perspective article to this one. And they say that the researchers, quote, offer convincing evidence that female budgerigars modified their mate preference in favor of trained males after observing them perform complex foraging tasks. However, it's not entirely clear that they appreciated the box opening as a sign of intelligence. Right. Quote, the fact that females lacked the opportunity to perform the task themselves suggests that they may have had little basis for understanding the exercise as a problem in need of a clever solution. They might have instead attributed male success in opening the containers to superior physical strength.
Or again, can I just throw in?
Just where the seeds were.
Where are the seeds?
Look,
Who has the food?
yeah.
Look, if you trained one to open a box, no seeds, and you train another one to like lift a tiny bird dumbbell, no seeds, and the third bird just laid back and like mysteriously from a little funnel above, seeds dropped down and covered that bird in seeds.
Right.
I can tell where she's going.
Right. So the idea here, at least, is that this is an interesting initial finding, and that there's the workings here of a good mechanism to test sexual selection of intelligence. There needs to be some adjustments, most likely, especially based on the University of California, Irvine group here. It seems like they need to make sure that the female understands that there's an intelligence factor in solving the puzzle before her making that decision can mean that.
I guarantee you, I could put a picture of a cat in that cage, but drop a pile of seeds in front of it. I know where she's going. She's going to the pile of seeds in front of the cat. It has nothing. This is like the worst designed experiment, in my opinion.
Well, we're not looking at the entire all the controls here, and so there were very likely. I mean, this is an article in Science.
Mm-hmm.
It's in Science magazine.
Full of people who like the idea.
They like the idea, but at the same time, there's a bunch of smart people who are going to be asking questions like you're asking. And so I'm assuming that some of these things you're bringing up were addressed in the paper. From the abstract, it says they had control experiments in which females observed males with free access to food.
Mm-hmm.
And there were others in which females observed female demonstrators solving the problems.
Were they still attracted?
No.
I mean, that's
no, the preference shift didn't happen. They already chose their favorite.
Okay.
Yeah, so the idea is obviously there's lots of layers we're not seeing here. This is not just a simple science experiment. This is a full research experiment. But what the UC Irvine people are saying is that there's interesting implications here. And I think it merits future research. And it's pointing a certain direction, but we need to rule out the idea that physical strength or just availability of food has something to do with it, which you can do.
Which is good. And I mean, I like this, but it's also it does also say that anything that's an advantage to survival, whether it's being the biggest or the smallest, the strongest or the fastest or the whateverest. All of these things they're all strategies for survival. And if you're gonna, lay your egg with one bird this season, you want the one that's got the best strategy for surviving, or the
right. I mean, and it makes sense if you think about there's the whole like Maslow's hierarchy of needs thing for humans. It's similar for animals. They're going to care a lot more about who can get them food than who has the right song, in theory, which is what this is actually testing, right? Yeah. Is if intelligence helps get you food, that should trump the ability to sing a beautiful song or look beautiful. And that's, I think, really, we're using all these fun words to describe it, but that's really what this study is about.
And also, may I say that knowing a little bit more about the depth of the study, I'm a lot more in favor
of it. Excellent. They have controls. They've got it under control.
Fantastic.
Of course.
Study this.
Is it time to go to a break?
I think it's time for a break.
I believe it is time for a break as well, everyone. Thank you for listening to this first half of This Week in Science. We will return in a few moments with more This Week in Science. After this short break, stay tuned.
Can't explain things you've heard with more than intuition. A line breeze that shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need.
Thank you, everyone, for listening to TWIS, for watching TWIS, for being a part of our community and enjoying science with us. We appreciate you taking this journey with us on a weekly basis. In order to keep the show going on a week after week basis, we need to ask for your support. This is a listener-supported show, and it's listeners like you who help keep this show going. You help support all the things that we do. And if you're interested in supporting TWIS, let me show you a few ways that you can make that possible. First, if you head to twis.org, that is where you find all the ways and all the things TWIS-like for helping us out. If you would like to order our This Week in Science Blair's Animal Corner 2019 calendar, there is a link on twis.org on the main page. There are just a few left. Really, they're almost gone, almost all gone. So get yours now. Get one for a friend. Also, you can support us through donations. And so if you scroll down that main page, there's a yellow button. Click on it. It'll take you through a PayPal interface where you can choose the amount of your donation to our program to help pay for things like our bandwidth and our new cameras and other things that we need to keep the show going. We also have a Patreon community. So if you click on the Patreon link, that will allow you to support us in a monthly ongoing fashion. Click on the red button once you get to the patreon.com/thisweekinscience page and choose the level of your support. You can choose $10 a month and we'll thank you at the end of every show by name. $15 a month and we'll send you an MP3 disclaimer for your voicemail. That sounds like fun, doesn't it? And there are many more options for you to choose from. Whatever fits into your budget, it's just one donation a month. The $10 a month donation is really the same as buying a cup of coffee once a week. And that's a great level of support to start at. And you said it, forget it. Well, don't forget it. Be a part of the community. That's the best way to do it. We also have a Zazzle store. So if you're looking for something like a t-shirt or a hat or something with a TWIS logo on it, that is where you go. So back to twis.org, click on that Zazzle link and find all the TWISy items of your dreams. Also at twis.org, the one last way that you can help us out is by subscribing. By clicking the subscribe button, it will lead you to links for YouTube, iTunes, or Google Play, where you will be able to subscribe in the method of your liking. If you're not interested in those three places, well, we are other places. Just look for This Week in Science in Pandora, in Spotify, in Spreaker, in Stitcher, in TuneIn, all over the web where good podcasts are found. And your support in all these ways is what keeps our show going. So I thank you for your support. We really could not do the show without you.
Can't explain the things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scatter plot. First assumptions were correct. Let's prove the rest. It's hot. The methods of hypothesis and patience.
Oh, the mark. I see that. Let me say thank you.
Put on a pair of goggles and go looking for the things I couldn't see.
And we're back with more This Week in Science.
Yeah, we are back, and I have a wonderful This Week in What Has Science Done for Me Lately. It's a bit long, but it is wonderful. It's a story, so I hope that you are ready. Kick back and relax and listen up. Hi, Dr. Kiki. This is probably too long for you to read on the show.
It's not, obviously we're doing it right now.
I unfortunately talk a lot. This gets transferred into me typing a lot. This is my What Has Science Done For Me Lately story. Hi, my name is Ash, and I'm a high school science teacher in Sydney, Australia.
Woo,
I've been
Ash.
wanting. I know. Go, Ash. You're melting the brains, forming the brains of the next generation. We're making this longer. Okay.
Sorry. I'm sorry.
Yes,
I'll start
I
to
have been
myself.
wanting to contribute to What Has Science Done For Me Lately for a long time. However, not being able to quantify what lately actually means, nothing has really stood out. As I try and communicate science to whoever will listen, even beyond my time at school, Blair's 700th episode note inspired me to write in as it made me think about the way I teach. When I get a new grade of year 7 students, first year of high school in Australia, I always ask the class to draw a scientist for homework at the end of their first lesson. Rest assured, 90% of the time, their image is Einstein, Esquire. Sometimes there's the odd girl or Professor Frink from The Simpsons. After they're finished, similar to Dr. Kiki did for Blair, I ask students if they've ever asked why about anything, solved a problem for themselves, worked out how to fix something, even tried different strategies to pass a really hard level on a video game. If so, they have engaged with at least a part of the scientific method, and they themselves are scientists. As they progress through high school, they are going to learn the skills to be even better, more informed scientists, of which they are extremely enthusiastic about and eager to learn more. They repeat the activity, drawing themselves as scientists, and everyone is keen as beans to get learning. My new phrase.
Keen as beans.
Keen as beans. There we go. Unfortunately, there is some correlation between puberty and a demise in this enthusiasm, which I think kicks in when teenagers realize they know everything. Fortunately for most, their enthusiasm is rekindled in about grade 10 when they start thinking about their future. But what has science done for me lately? In 2017, myself and 3 other teachers were selected for the Australian Science Teachers Association to go on Bush Blitz, a program funded by the Australian government and various private organizations to send about 20 specialist taxonomists into the field of remote areas of Australia for 2 weeks with the goal of discovering new species and recording range extensions for known species.
Wow.
As a biologist, this was really amazing. It was an amazing experience because the only field work I did in my undergraduate degree was collecting rocks for one of my lectures of research. There were a few new plant
I
and
would
some
love
animal
that.
species found, as well as apparent new spider genus, but I haven't seen a paper published on it yet, so that may have fallen through. To sit around a table with all these specialists in their field, enthusiastically talking about their discoveries of the day over dinner was incredible, and I wanted to spend as much time with all the scientists, absorbing all the knowledge that I could. This experience inspired me to be a more involved citizen scientist, using various app-based technology to help with the recording of species in my local area. Furthermore, it increased the cache of skills I have to teach my students and has helped me engage them with their local environment, giving me the necessary skills to head outside and confidently teach how to record abiotic and biotic data, catching and identifying spiders, insects, and recording sightings of larger animals with the aim of creating long-term surveys, monitoring the health of our local ecosystem as we have an airport opening up 6 kilometers from us in 2026. The experience also inspired me to return to university, and I'm currently doing my master's in zoology, entomology part-time while working. I'm loving all the skills I'm learning and have come up with some research that I may look to undertake in regard to ecological education in the future. Communicating science is my life, and I am a better educator because of TWIS. My students love hearing all the things that I have learned from your show and often diverging into a rabbit hole of tangents that have nothing to do with the curriculum. Some stories have even helped me to improve my results because you direct me to extremely current research in my assignments that my lectures want to then discuss with me. Maybe when uni is all done, I'd be able to collaborate with Blair as a colleague if I'm lucky enough to score a job as an educator at Taronga Zoo in Sydney. In summary, science, it has made me who I am. Science, hockey, and music are all I really talk about. And whenever conversation hits a lull, I start a sentence with, "So, have you heard about?" And we'll start talking about something that I have read or learned from this show. It keeps dinner gatherings with my relatively conservative family interesting. I know this isn't what you usually read out in this segment, but I just wanted to contribute to the show. Good science to you all. Ash, if you want to see my students loving science, check out at Ash64. That's ASHSIX and the number 4. Or hashtag science with Mulcahy. Sorry. Hashtag science with Mulcahy. I'm looking over on Instagram. So he's got an Instagram and a Twitter. And he's got an Instagram that you can check out the work he's doing out there, everyone. I love this. Ash,
Cute.
thank you. And Blair, you have a colleague, a future colleague.
Yes.
Ash, it was, I know, it was fantastic. It was long, yes, but I think well worth it. And I am so excited to hear what science has done for you. You're educating people in science. You are furthering your education in science. You are reaching out into the community and you are affecting the future. And Ash, that is just music to my ears, even though I don't have hockey in there anywhere. But thank you.
Ash, you'll have to keep us posted after you finish your master's degree. Can't wait to hear all about that. Tweet at us, write in again in a year. I don't know.
Yeah, let us know. Yes, I agree. Absolutely. So anyone else out there interested in writing in with your story? What has science done for you lately? It can be a longer lately. It can be a quick, short lately. It could be a haiku. It could be a sonnet. It could be a song. What do you have to tell us about science in your life? Email me, Kirsten, Kirsten, at thisweekinscience.com, or send us a Facebook message on our Facebook page. It's This Week in Science on Facebook. Justin, tell me your stories.
All right. So one of the rules of Fight Club, not talking about Fight Club. One of the rules of This Week in Science is not reporting on cancer cures. And it's not because we don't want people to know about cancer cures, but because just about every story about cancer cures is heavily caveated.
Right.
With statements like, I'll try to give you a rundown, a synopsis of a typical cancer cure story. This may potentially someday lead to a pathway that opens a door for what might lead to a new possibility in the search for, and it keeps going like that. But the following cancer cure story that I'm about to bring you is slightly less caveated than most.
Slightly less. Thank you for that.
Slightly less. So yeah, go ahead and count the caveats as you like. A new drug shows potential to halt cancer's cell growth by stunting the surgical clock. The findings from scientists at USC Michelson's Center for Convergent Bioscience and the Nagoya University's Institute for Transformative Biomolecules looked at turning the body's circadian rhythms against cancer. Their study, conducted on human kidney cancer cells and on acute myeloid leukemia in mice, was published today, January 23rd, journal Science Advances. So disrupting circadian rhythm is known to harm health in humans, apparently in rhesus monkeys as well. Same is true for the circadian clocks of cells themselves. So if you disturb the circadian clock of cancer cells, they theorized, you could potentially hurt them, or maybe even kill them. Caveat. Scientists found that a molecule, GO289, targets an enzyme that controls a cell's circadian rhythm. This drug protein disrupts the function of 4 other proteins that are important for cell growth and survival. Quotey voice of Steve Kay, who's director of Convergent Biosciences at USC Michelson Center. In some cancers, the disease takes over the circadian clock mechanism and uses it for the evil purpose of helping itself grow. With GO289, we can interfere with those processes and stop the cancer from growing. So GO289 can mess with cancer cells' hijacked circadian clock, slowing its cycles. And it can do so apparently with little impact to the healthy cells. So they Yeah. And its
So you
initial
wouldn't get that
interaction.
schizophrenia or depression or anxiety like the macaques did from earlier.
Right. So its
No.
initial interactions with human bone cancer cells, GO289, appeared to slow tumors' circadian clocks as it targeted a different enzyme, CK2. To see if it consistently hindered other cancers in the same way, scientists then tested it on human kidney cancer cells and on mice with the acute myeloid leukemia, and they found that GO289 specifically affected cancer cell metabolism and other circadian related based functions that normally would enable the cancer to grow and spread by the hijacking behavior. So
That's insane that you can target the circadian rhythm of the cancer cells specifically.
Yeah, and so it's I think what they're doing is actually targeting the cells, the normal cellular circadian rhythm, which is triggered molecularly. Chemically molecular, however you want to put it. And the fact that the cancer cell relies on the cell's native circadian rhythm and is then changing it, and that is what's affected, right?
It's very cool.
And then this is the ending, a little bit more Cody voice. Kay is optimistic about the findings. This could become an effective new weapon that kills cancer, he says. So it's unfortunately a thing with all of the cancer research. They seem to get it's almost like they get a thesaurus of caveats, a caveat of sorts, with which to pepper anything that they're talking about, so as not to create too much hope, I suppose. But it is a fascinating new way of targeting that heretofore I have not heard of.
Well, and it seems like even if it doesn't kill the cancer, it might slow the progression down, which would also be good. There's lots of super aggressive cancers out there that if you're given a little bit of time, you could treat it potentially.
Find a path towards a possible cure from a different avenue if it were to arrive by slowing down the progression of the disease. Yes,
there
exactly.
Yeah, and if you just get it to the cell to think it's time to sleep and not time to grow and divide, you've effectively stopped it. So,
because I mean, this is of course what we talk about. This is what cancer is accelerated growth. So a normal cell's growth pattern is much slower than cancer. So if you slow it by 50%, it's probably still growing at a rate that would be somewhat analogous to its normal healthy behavior, but is a massive reduction in the amount of growth that is taking place within the cancer. So, fascinating technique, fascinating way of finding that technique and applying it. And I really would like them to be successful. As with I wish that for every
right. Be successful.
There's not a cancer cure that I'm like, I hope they don't win. But you know, this already got some preliminary good data out of it.
There you go.
Fantastic data. Fantastic. Speaking of other health news, there was some research. Oh yeah, have you had your gums checked recently?
I'm going to the dentist next week.
Good.
Got a new dentist I had to go see soon.
Yeah, well, you might have them check for I can't even say the word. I can't speak today. My brain just turned off, everyone. Powering down, computer Kiki off.
Can't we all have Kiki's powered down brain? I would take it.
Totally take that over there. Or Justin full blast. It's gotta be great. Gotta be awesome in there.
Well, you might want to you might have to, put up with some other things, but you wouldn't have to put up with periodontal disease. There we go. Well, it appears some researchers from the University of California, San Francisco, and a company called Cortexime have been looking into a bacteria called P. gingivalis, which causes periodontitis, periodontal disease that is an infection, bacterial infection in your gums that destroys them and can make your teeth fall out.
Right. Yeah,
don't
like
want that.
the receding gums and the
Yes. Well, additionally, they have found that there is a link between P. gingivalis presence in the gums and the bloodstream and your brain. And there is a link between P. gingivalis and Alzheimer's disease.
Everybody go to the dentist.
Everybody go to the dentist.
It's like a whole nother reason. So many people avoid the dentist.
Yeah. So,
do you want to lose your memories? Go to the dentist.
yeah. So these researchers have been searching for these infections, things, this idea that, the immune system is involved in Alzheimer's disease. And so there may be infections that spur some, but not all of the cases. And if there are infections, how can we stop those? And we can stop a certain percentage of cases of Alzheimer's disease. And so they have been looking for signs of a link there and discovered this particular link. They found DNA from the bacteria in the brains and cerebrospinal fluid of Alzheimer's patients.
Wow.
They also found bacterial enzymes called gingipanes. I like that name. Gingipanes. They destroy brain cells, not your friend. Cause lots of pain. And anyway, they watched these infections in mice and they saw neurodegeneration in the hippocampus and buildup of amyloid beta plaque production and inflammation. So they put together, and this is the end of the story, that they created a molecule. This is what this Cortexime company is good at. They created a molecule to block gingipane enzymes. It reduced the amount of bacteria in the infected mice, stopped amyloid beta plaque formation, and reduced inflammation. And this compound, which is called Core 388, has passed human safety studies and is on its way to clinical trials.
So fantastic. However, the first question I would have is the directionality of this. So, if you attack it from, this is a mouth bacteria, and is because it affects your teeth. It's a mouth bacteria, and it begins and starts there, and this is the origin of it, and then it goes out and causes problems elsewhere. Then, fantastic. But if it's a spinal fluid initiated enzyme or bacterium that then finds its way over to the mouth and infects the mouth,
no. I believe it starts in the gums. The gums are the pathway to the nervous system.
But couldn't the gums just be an accessible place for bacteria that could be like, it could be in the gut, or it could be somewhere else in smaller form? Really accentuates there, but not necessarily the source of it. I mean, the idea would be you can wipe it out from the mouth, but then this bacterium is still causing the release of the enzyme or what have you, that's causing the effects that get into the spinal from some other source. I mean, that's just
yeah, I suppose it's possible, but what we know about periodontitis at this point is that proper dental hygiene does make it go away in terms of what's in the mouth. So if we know about that, if we know that, occasional deep cleanings and flossing and twice daily brushing and all that kind of stuff prevents this disease in your gums, then I think it makes sense that that's where it starts.
Okay.
So but I could see where you're saying that you could just be keeping it at bay if it's coming from somewhere else, and you're just keeping it out of your mouth through that process.
Right. And the other thing is like that you always have to be careful when you start talking about dental hygiene in the terms of eliminating, because I guarantee there are people with bacteria in their mouth that either cause plaque or cavities or have some other maybe, or nothing at all.
But it's not to do with eliminating.
But that are out competing that bacterium. Yes. So if you don't have it, if you don't have this P. gingivitis, whatever it is, if you don't have it in your mouth now and you cleanse your mouth completely, you may have created a perfect landscape for it.
Yeah.
No,
All right. Yeah.
No, it's absolutely right. Yeah.
I had a conversation like this with my dentist a long time ago, where they're like, you have a lot of plaque buildup, and it's a bacteria that causes this. And I was like, okay, but I don't have any cavities. And they go, oh, well, that's a different bacteria that causes cavities. I'm like, so why would I ever give up the plaque ones where you can go in and scrape it off once in a while? Well, otherwise, the other one, if I cleanse it, that one might move in. You got to drill everything out. Like, that's ridiculous. Like, I want you to do whatever you do. Do not eliminate my plaque-causing bacteria, because I don't want
that's
to move
it's a population. It's a population. It's an ecosystem. And the issue is that if one of these gingivalis, if the gingivalis species is allowed to overrun for whatever reason, poor dental hygiene, whatever it happens to be,
too much,
it can lead to, or too much. Yeah, it can lead to the periodontal disease, which can lead to an entryway into the bloodstream. And then even if you fix it at some point, you still potentially have those proteins and enzymes in the body for a while that can cause trouble.
So side story, or side study
things
that could come from this. Let's study people with Alzheimer's and compare their dental hygiene to that of the general public. And if it comes back that people who get Alzheimer's,
I thought you meant people after they have Alzheimer's disease.
Yeah, people who get Alzheimer's disease, compare their dental hygiene. Going backwards, certainly not forgetting to brush, but going backwards. Compare their access to dentists and dental insurance and dental hygiene, or however you want to quantify this, to a subset of the population that has never gotten it or doesn't get it, and that would be very interesting, wouldn't it? I think
it might be. It might be. But, I think it's great that they've got a molecule. They've got a molecule now where maybe it'll work and we'll be able to block that protein and solve a lot of that problem. So you'll be able to have your poor dental hygiene and not worry about it.
As well.
Yeah, it's great. There is another Alzheimer's study out this week from the University of New York, Buffalo, looking at Alzheimer's disease and influences in what way that we could possibly reverse memory loss. They have a paper that they just published identifying epigenetic factors that contribute to the memory loss. They found ways to temporarily reverse them in a mouse model. And it's a very exciting study, other than the fact that they've cured Alzheimer's in mice and not in people. So they gave mice a familial Alzheimer's disease, which means that it's genetically carried and passed on from generation to generation. And they looked at epigenetic changes occurring in the mice. They discovered that there's a loss of glutamate receptors that are critical to learning and short-term memory, and found that many subunits of glutamate receptors in the frontal cortex get down-regulated and disrupt excitatory signals, which impairs working memory. So if you have bad working memory, you're just not able to keep things in your mind to be able to remember things.
So
anyway,
can I slow you down real quick? I'm going to slow you down just for a second, and just maybe jumping ahead of the story, and I apologize. That's fascinating to me, because I've always considered things like Alzheimer's to be the destruction of the stored memory.
It's not
but you're
necessarily
talking about
communication.
Yeah, you're talking about access. So there's a difference. There's a difference which never occurred to me. There's a huge difference between an access to a memory and the actual storage of it somewhere, the imprint of that memory somewhere in the brain.
Wow. Yep. Exactly. I'm so glad I was able to help blow your mind there this evening. That's awesome. Anyway, they found that that the loss of the glutamate receptors is part of what is called repressive histone modification, which gets elevated in Alzheimer's disease. And this is something they saw in mouse models and in human postmortem tissue. They looked at this abnormal modification, the diminishing glutamate receptors, and they ended up and they found a bunch of drug targets that have to do with enzymes. And so they gave the animals an enzyme inhibitor and saw the rescue of cognitive function, which they confirmed through evaluations of memory, recognition, spatial, and working. And they say that they were quite surprised to see such a dramatic cognitive improvement. And they saw the recovery of glutamate receptor expression and function in the frontal cortex. They looked at it only for a week. So they saw improvements for 1 week, and they're going to see if they can create compounds that penetrate the brain better and are longer lasting into the future. And so that's where they're going. They've created an enzyme that can restore memories for Alzheimer's disease in mice.
Right. And every time I hear a thing that cures something in a disease, I wonder if it could just make my undiseased brain stronger. And if I could do this, if I could take this, could I remember stuff that I haven't thought about for a long time just because I shut down the access to the memory?
Or something that you lost after a night of heavy drinking, perhaps.
Well, there we go. Yeah.
I'll never get that back again. You can only lose that once.
You probably don't want to get that back ever again. Right. Tell me another story, Justin.
Oh no. Do I have more? Oh shoot, I hope I have more. Okay. Oh yeah, this is a separate skeleton suggested to be from disparate early hominin species are, as it turns out, sourced from the same species, a team of anthropologists have surmised. The research appears in a special issue of the journal Paleoanthropology, which I wish somebody would send me. And it's part of a series of articles that offers a comprehensive accounting of Australopithecus sediba, hominin species discovered in South Africa back in 2008. So they had a couple of specimens here, very complete specimens, and they were over a past decade, researchers have been piecing together these skeletons, and basically the culmination of this work, a series of 135 fossils, and I think 9 different papers are in this paleoanthropology coverage. And it covers, in some detail, very specific limbs and other aspects of the vertebrae and the skull. And there's a fantastic video of this. If you can click on this story and do a screen share, they actually have a animated skeletal walking, a bipedal walking. So this is one of the other things that they've totally also come up with, is that this was a bipedal walker, you know. This is, and again, this is a 2 million year old fossil collection from the Witwatersrand in South Africa. That they say this sediba was both adapted to terrestrial bipedalism and also spent significant time climbing trees, either to go and find tasty things to eat in the trees, or to find ways to avoid being tasty treats for predators around. Quotey voice of anthropologist at New York University anthropologist Scott Williams. This larger picture sheds light on the lifeways of
nice work.
sediba, and also on major transition in hominin evolution that have largely ape-like species, included broadly in the genus Australopithecus to the earliest members of our own genus Homo. What's also interesting is they say this is clearly a distinct, disparate species from our own, but is also connected to another species that another hominin that they had been looking at as potentially a separate species, but is not a direct ancestor, even though it shows a lot of morphology similar to what ended up in a perhaps common ancestor. And they had, I guess what they thought had been 2 disparate species turned out to be a juvenile and adult, and so they sort of were able to track morphology over time and realize that this is what this other one morphs into, and the morphology alone wasn't enough. Another sort of ancient human-y story there's a Neanderthal story out, just kind of interesting because it's in one way it's very contradictory. They're both. It's a new discovery that sort of is finding that Neanderthals in southern Iberia were replaced by modern humans much longer ago than previously predicted, because there's a site very close to there that had been the longest holdout of Neanderthals. So they're putting this at the site was approximately 44,000 years ago. The current humans replaced the Neanderthals on the site, but the other site that's really not too far away, Neanderthals persisted to 32,000 years ago as the dominant hominin. And all these are sort of contradictory in the whole, like, when did the current humans replace the archaic humans in the region? The sort of sub story here is that it looks like it's possible that we cohabitated the same lands for 10,000 years or more. That this was, and it's not so much a humans in, Neanderthal out scenario, but Neanderthals to the north, Neanderthals to the east or west, and current humans to the east or south or what have you, for 10,000 years of potential cohabitation and intermingling. So a lot less of a direct replacement. Even in some of the commingling cohabitation scenarios, they always try to accelerate the event with lack of the hybridized next generation of human and Neanderthal being reproductively less efficient, or
Right.
The idea that we had many thousands of years to sort of overlap a bit is really fascinating, and sort of does challenge a little bit that replacement scenario.
I don't know. I think, yeah, I mean, that scenario is basically, that just is going away.
Yeah.
Let's take this old idea, this story that was told to excite us about Neanderthals. They were here before, and then humans won, and we kicked them out. You know, the story of humans. We won. We survived. The sediba, Neanderthals, Denisovans, Floresiensis, all of them, all of the ones that we're learning about are telling us this very different story, this nuanced story, and this kind of information. I think it makes sense, especially considering how much Neanderthal DNA is mixed in with humans.
What do they call it now? A braided stream, right?
A braided stream, exactly.
Yes.
And also funny, interesting to note that in thousands and thousands of years of Neanderthal and current human, and a hybridization element in between, not once is there any evidence of a wall being built between the two.
But it is an interesting question, though. Culturally, what was shared? Was there a common way of communicating? If humans and Neanderthals were in the same areas for that long, they overlapped. Were they speaking the same languages? Were they learning each other's languages? Were they trading goods? Were they, I don't know, were they sharing? Were they learning? Or were they just completely tribal and shunning each other?
Didn't we have a story last year about Neanderthal art?
Yeah. So all of the French and Spain ancient cave art that we attributed to the earliest man, current human artwork actually turned out all to be Neanderthal. So it's very likely that if you were to put it into some sort of weird modern terms, very likely that the Neanderthals were the Etsy crowd, and the current humans of the day were likely the real estate agents. I feel like that's kind of how we were going to be best represented.
Sure. Yeah. I like
it
it.
Let
was
me
sort of
let
like,
me let me show you this cave over here. It's a very nice
Yeah.
cave. Doesn't have much
Yeah.
of a draft, but you know it'll pull the smoke out. It'll be all right. You can light
It's
your fires
muskox
right here.
adjacent, and
one was very real estate aggressive. And the other one was very much sort of into a slower paced life where art was the, sort of the mainstay. Is sort of what the picture that we're getting from the
Interesting.
ancient past.
Well, thinking of things from the ancient past, I've got some quick science news stories here at the very end of the show. Quick and dead. Let's. Researchers have been drilling into Antarctic lakes, Lake Mercer in the Antarctic. They drilled and have come to Lake Mercer and gotten a sample. They found little tiny dead things. They found dead tardigrades, dead crustaceans, things that looked like dead squished spiders. Yes, they have found the vestiges of many organisms, and this is first reported last week in Nature Online. And the SALSA team is the team who is responsible for this. SALSA, known as Subglacial Antarctic Lakes Scientific Access, have been digging into Lake Mercer to see whether or not anything was there, because they've dug into lakes before, not finding anything. This one, however, they found a bunch of dead things, which indicates that possibly what happened through the past is that different glacial periods, this lake has been open to the air and actually had a thriving ecosystem. It later froze over. Things died in there. Maybe it opened up again, allowing things back in, and then froze back up again. And maybe it's got a freeze-thaw cycle that goes on and on over eons, and that explains the lack of life that they've discovered, but the preponderance of little frozen dead things.
There you go. Those tardigrades might even wake up if you thaw them out.
So it's interesting, and it also makes me wonder what they're looking for. I mean, there was a receding, and I don't know, did I do the story last week? I might not have done the story, but there was a receding glacier that they found 2 new species of fungi under.
Cool.
That had been surviving
Buried
under a glacier and was reproductively active at 0 degrees Celsius.
Amazing.
Yeah, which also like was industrial application, except for most of our industrial processes are heat oriented. But this is one of these things where it's away. Quick, I'm not actually going to do this story.
I just have one. I have one quick. One more quick story, and then it's your turn. And then it's your turn, so I can prep some things for the end of the show. Gene Drive. We've talked about Gene Drive before on the show, and it's been very successful in mosquitoes and putting a gene into the mosquitoes or taking a gene out of the mosquitoes, but basically driving the gene out to leave mosquitoes in a lab reproductively sterile. It's been effective. Just 100% of the population of the lab mosquitoes was wiped out. However, researchers have been working on this in mice and haven't had, or other animals, and haven't had quite the same success. But now UC San Diego, who we've reported on before, working in the Gene Drive area, has had some success in mice and working with a gene for coat color, which wouldn't matter whether or not it got out of the lab necessarily. They swapped in different chunks of DNA that caused a DNA cutting enzyme to be active in different cells. And so when it was active in male germ cells, the ones that create sperm, it also created large deletions rather than converting to normal copies of the genes. And they had also the enzyme active in female germ cells before the DNA was repaired with a copy gene drive payload. However, they're working it up. The efficiency, as opposed to the mosquito 100%, their frequency was about 72%. And while this is still not where they want it to be, this frequency of inheritance for this particular gene for coat color was much higher than normal Mendelian inheritance. And so, of course, in their paper, they entitled it Super Mendelian. So that's going to be my superhero nickname.
Super Mendelian.
I'm Super Mendel. That's right.
Will you carry around pea pods?
Possibly.
Throw them at people?
Very,
Hiya! Hiya! Hiya!
very large ones.
Okay, here you go.
Yeah. So eventually, this is the research that's going on in this system. The authors state the goal of using the system to put together interesting combinations of mutations seems to be within reach. Excuse me. But there have been some suggestions of using it in populations of wild mice to eliminate their ability to carry some diseases or as part of an eradication effort where rodents have been an invasive species on remote islands, for instance. And that's not going to be ready for game time anytime soon.
Mm-hmm.
But this is a good step along the way. If we think of gene drive as good, and so good step in it. Yeah. Anyway, gene drive. Okay, Justin, tell me your story.
Not to be left out for saying Mendelian. Results of Mendelian randomization study indicate that accelerometer-based physical activity appears to protect against the risk of depression. Basically, on average, Cody voice, on average, doing more physical activity appears to protect against developing depression. Any activity appears to be better than none. Our rough calculations suggest that replacing sitting with 15 minutes of heart-pumping activity like running, or with an hour of moderately vigorous activity, is enough to produce the average increase in accelerometer data that was linked to lower depression risk. The one thing I'm not even really going to go into this story. The one thing I'm going to point out is they did not in their study incorporate anybody who was rowing a boat under a slave-like condition, or anybody doing forced labor, or anybody who's been doing physical labor as their career job, which they foolishly fell into right out of high school, as opposed to getting a higher education and have been stuck with being physically active for the last 40 years. So I'm just going to say, yeah, for those of you who maybe have not exercised much in your lives, maybe that could divert some depression from an otherwise unphysical life. But for somebody who might be perhaps physically involved in their day-to-day, every day, I don't think that an additional physical activity is going to help. These things are all subjective. For those people, maybe taking time off of physical activity could reduce depression. You really have to be careful when you're making these overarching claims about things that take place in comparison that you're talking about.
The one thing this makes me think of, though, is that the whole kind of fad with getting your steps in, wearing a Fitbit or an Apple Watch or whatever it is, there might actually be something to that, because the people who are saying I got to get my steps in are the ones who aren't moving around very much the rest of the day. Okay. So they have to make sure to budget the movement, and that might actually help. Very interesting.
However,
stress. If your thing keeps telling you, go walk now, you haven't walked enough. You need to walk some more. Ah, but I just did walk. No, walk some more. Ah, okay, Fitbit, I'll do what you say.
There is an idea that these Fitbits and other health devices don't actually help in terms of mood management.
Well, because you might actually get anxiety about not closing your rings or whatever. Absolutely. Speaking of physical activity, I have one last story for the end of the show that is very interesting, but very short. And it is that an animal has been shown to be doing something hitherto unknown. Is that a real word? No, it's not.
Yeah, you just said it.
So,
therefore it's a word.
There's a few individual animals that we have seen walking on water: spiders, water striders.
That awesome Jesus lizard? What is it?
Yeah, the basilisk, or the Jesus Christ lizard. And so we know how they do it. But now a gecko has been seen running on water, which is really weird because we know how those other animals are doing it. The spiders and bugs are doing it by surface tension on waters because they're very light. And then the basilisks are doing it by size and strength to push themselves above the water as they move. And this gecko is in between in size. So we have no idea until now how they're getting this done.
Like the grippy paws, but not the really big surface
related.
area.
So not only did we not know that the geckos could do this at all, but then once we found out that they could do it, we're not sure how. So this is a study from Rockefeller University, and it turns out they use surface tension, but they put them in soapy water and they killed surface tension, and then they couldn't move. So surface tension was definitely part of it, but it wasn't all of it. So they also are physically creating air bubbles, generating force, just like the basilisk does. But there's something else. It's actually also hydrophobic skin, which is kind of related to this weird sticky thing that they're doing too. Yeah. But the hydrophobic skin repels the water and enhances their ability to stay above the surface. So it's actually 3 methods in one, 2 we've known about, and 1 brand new one. So the idea is you could potentially make a medium-sized robot that could move on water in order to do search and rescue in floods and things like that, because swimming is actually way more energetically difficult and slower than moving on top of the water, as we all know. So there is some practical potential uses for this, but also it's just really cool.
Yeah.
I love that it's not just the normal things that we have seen previously, but it's this hydrophobic ability and air bubbles, and that's awesome.
Yeah. It's all the things.
Oh, geckos, you're just so cool.
So cool.
Geckos.
So cool. Oh, and I forgot to mention that story comes from a Kirsten Sanford. Oh, Dr. Kiki, you sent it to me. That's right.
I thought you would like it.
I loved
thought
it.
You would enjoy it.
I loved it. In fact, I'm taking that to work with me for sure.
Awesome.
Yeah.
Yes, your animal friends will appreciate understanding their other animal friends at the zoo. All right, everybody, we have done it. We've made it to another end. It's another end. We're here at the end. Isn't it beautiful, my friends?
This is the end.
Yeah, we are here. And I want to take this moment to say thank you to all of you who listened for the whole show. Or if you're just here because you listened to part of it, thank you for being here right now. Thank you to Identity 4 for helping to record the show every week. Thank you to Fada for helping with show notes and show descriptions and social media. Amazing. And thank you for helping in our YouTube chat room. Gord McLeod, thank you for helping to moderate in our main chat room. For our YouTube donors, thank you for donating today during the show. I appreciate your donations. And I would like to thank my Patreon sponsors. Thank you to Paul Disney. I scrolled down. Richard Onemus, Ed Dyer, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Massaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Aiden Jeff, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Corinne Benton, Sue Doster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zuknarek, Ashish Pants, Ulysses Adkins, Sarah Chavez, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim Drapeau, Craig Riley, Sean Lamb, Ben Rothig, Steve Lesseman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and E.O. Thank you for all of your support on Patreon. And if any of you are interested in learning more about Patreon, you can find information at twis.org. Click on the Patreon link if that's what you're looking for. Remember, you can also tell other people about TWIS. And on next week's show, we will be back to talk about science. Live online, 8 p.m. Pacific time on Wednesday night at twist.org/live. You can watch and join the show, the chat room live. We can talk with you. We can see what you're doing if you're here. But if you're not and you can't make it, don't worry. We love the fact that you are subscribed to the show and can listen and find past episodes at youtube.com/thisweekinscience or twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have the mobile type devices that are out now, you can look at TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science in anything Apple marketplacey.
Are you looking for more information on something you heard here today? Show notes are available on our website. That's right. You know what it is. It's www.twist.org. That's TWIS.org, where you can also make comments while you're there. And you can start conversations with the hosts or other listeners.
Or you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, which is spelled TWIS, somewhere in your subject line. Otherwise, your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We will be back right here next week, and we hope you'll join us again for more great science news.
Oh, but remember, if you've learned anything from this show,
It's all in your head.
It's right here. I think I've got
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science.
This Week in Science.
This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just get understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the cause it's This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just
listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
Remember, it's all I was waiting for.
I know.
It's all I was waiting
You said remember at the beginning of the sentence, which was confusing.
for. Like, wait, it's not the right
That's
order.
not my cue.
I have a cue and I wait for it. I wait for the word. Sometimes my brain is slow, like today when Robot Kiki has powered down. Yeah, I don't keep up as quickly. That's right. Tired, Kiki. Where's Justin? He needs to come back for our big, exciting news. He'll be back.
Does he know about the big exciting news?
Well, I asked him about it last week, and
Oh,
who
good.
knows? He might know, but he might not remember. Remember.
Right. That old thing.
Everyone, thank you. And there's Carolyn who is tweeted at me and said, "This is my favorite quote." Oh my goodness. "Your show is like vaccination for the brain against trendy ignorance."
Ooh!
Yes, it is.
Oh my god.
It's real information. And
I might
people
finally
asking
get a
questions.
tattoo, and it might be that.
It might be that, right? That's it. And that's why we need more people watching the show. Or not watching, listening to the show.
Consuming the show.
Yeah, isn't that quote so pithy, Identity 4? It is. Yes. Vaccination for the brain against trendy ignorance. Yes. Hi, everyone. Who did I not? There's a Justin. He's back. Did he put on his headphones yet? Your headphones on?
Yeah, and a
Headphones
blanket.
on.
It is freezing.
Cold.
Yeah.
Yeah, I was freezing last week and the week before that. It's a little warmer now. We've got a little bit of a warmer spell. It's like in the 40s right now. It's extreme.
Yeah, it's probably not even down to the 40s here. It's spoiled California.
It gets cold in the valley.
It does, but it doesn't get Portland cold ever. Yeah.
It gets cold here, but not as cold as
In fact,
it is in New York right
a
now. Oh my goodness.
Telltale sign of this was that I was visiting Portland once years ago. And of course, in California, I've never gone and put the like anti-freeze windshield wiper stuff in there. Like, why bother? You just put some water in there if you want to clean your windshield. Why not? And and I'm driving along, and my windshield is like got gunk on it and stuff. So I hit the little sprayer that sprays the windshield. Immediately turns to ice. Immediately turns to an ice sheet, and I have to slow down the car and pull over and have all the windows down and just try not to crash into something because I just created an ice sheet while trying to get some bird poop off my windshield or whatever it was. So yeah, like there's a difference being a Central Californian and never dealing with like frosty like conditions where you're
so cold. Yeah. Well, it's not Finland. Hey, Janiscu, that's cold.
That's yeah.
Minus
See,
10
now we're not even talking. Portland
Celsius.
cold is like summer for Janescu. Yeah.
Yanni, oh my goodness. New York is 51 degrees now. Wasn't New York City like -4 just 2 days ago? Oh my God. Ben Rothig is at 10 degrees with a wind chill of 2.
Oh, shit.
Cold.
Oh my God. My
121
shivering,
has
you
14
can walk into my shivering and warm
degrees.
up. This is what's crazy.
Burr, you guys are making me cold just thinking about it. Yeah, oh my God. I was outside in minus 12 degrees Celsius and it was so warm.
Ah,
That's not warm.
no,
That's
that's
not
not what
warm.
warm feels like.
That's not what warm feels like. No, it doesn't. Oh, the cold. Yeah, it was 4 the other day in New York City. I thought so, Noodles. That's great. And now 51. That's a rebound. That's a little what? What is the weather? What's happening? Oh, that's right, everyone. What's very exciting, our friends, let's see, comedy album, comedy album in the number one in the charts right now is Brian Brushwood and Justin Robert Young. All's Well, a Night Attack album. It is, I think, number 1 on Amazon. Yeah. And Night Attack is number 2. They've got number 1 and number 2. They're amazing.
Yeah.
They totally gamed that and that's bravo. Top 100 comedy albums. Brilliant. Oh yeah. What's the big exciting news? All right, everybody in the greater Portland area, or people who want to come to visit Portland, you better put your April 3rd on your calendars. We're doing a live show in Portland, Oregon. I don't have a location yet. But we will see. There were a couple of possible locations that we're looking at, but we will see. It is in conjunction with the local Science on Tap event, which is wonderful. Yeah, it means drinking and science. Yes.
Love it.
But we will be doing yes, it is the night before a science communication conference that I helped organize.
Yeah, and I'm doing a workshop at, so I'll be good and fresh after doing a tap room event
on Wednesday night. That's right. We're all going to be like, oh, and the conference.
Yeah, like, oh, you want me to talk for 90 minutes? Okay.
We're going to be good. We're going to be so pro.
Oh my God. It's great.
So pro. I know, Ben, I am live in Portland every week. It is true. But Blair and Justin will be coming to Portland. We're going to be on stage together in a theater doing TWIS. And so I got to figure out if we want to have any guests on the show to talk about their science, or we have to start figuring things out. We only have 2 months.
Let me just start by calling shotgun on the downstairs couch.
That's fine. I took the loft last time. I can take the loft again.
Okay, Blair's got the loft. Justin has the downstairs couch.
The loft. Is the loft better?
No, you don't want it.
Oh, no. Did I pick the wrong thing?
You don't want it. You definitely don't want it.
I thought I was getting out ahead of this. Apparently, I was.
See, Justin, I've been there. So
I've been there.
Oh, you have?
I've been there. I have. I don't know what the loft is. Oh, is that the studio? You mean the upstairs studio loft, or
way up? It's a crow's nest. It's loft.
It's a crow's nest.
No,
I like it. It's above my studio office.
Sorry, you called it, Justin.
I did. I'm sorry with it. Right.
April 3rd, Wednesday, April 3rd, Noodles. Yes, the date. Let me make sure, because April 4th and April 5th are the conference that I'm organizing. Yes, so the 3rd. It's a normal Wednesday, like our Wednesdays. It'll be, and I'm going to try to
live stream.
I'm going to try and do a live stream.
Yeah.
We will see what we can do. I don't know what tech abilities the theater will have, what tech abilities people will be able to have, but it's going to be a live show, so we will record it and it'll go out as a podcast, and if we record video, we will post video if we're not able to live stream.
Can I ask a question?
All right.
Because
Yes.
I've always noticed that there's activities taking place, but I don't always follow up with like figuring out if they like worked. I know we've attempted to do live streams multiple times in the past. How many of those worked?
It depends on the venue.
Yeah, it is dependent. It totally is dependent on the venue. Yeah.
Are there interwebby connections, right?
Mhm.
Yeah. I feel like the New York ones were pretty successful overall.
Oh,
And
the
I
New
feel like
York
those had
ones were
also a
amazing. We had
separate
such good help.
Yeah.
Was it Academy of Sciences wasn't going out live, or was it
The first, we were able to get the first Academy of Sciences show out live, I thought, or it didn't.
That was
Brian-
through like Facebook
yeah, through an
video
iPad or something.
through
yeah.
Periscope, one or the other. I
Okay.
don't remember. And then I think we tried to do that last time, but I think our location inside that Earth core, the Wi-Fi was not great.
The
So I
Wi-Fi
think-
was terrible, and
I
they
think
wouldn't
it
help.
died.
So
Yeah.
they're like, "We don't care about you wanting to stream." What is
Yeah.
that? What is
They're
that?
like, we have 5,000 people at this event that we're trying to entertain.
Yeah.
Like, oh, fair
All right.
point.
Fada's going to be here, so he says he'll help with tech. Yay! Awesome. Noodles, I know. I'd love to come back to New York. Should come back. Come
Yeah,
back
I
and
want to go back to New York. I freaking love New York.
New York's amazing.
Listen to you.
Yes.
I freaking love New York.
Yeah, I freaking love New York.
God, New York. Yeah.
Wait, what is she? Is she from Jersey or something?
I flew into Jersey last time in November. I took the train from New Jersey to Brooklyn, Had some whitefish. Great.
I
Said,
love
hey,
the
I'm
whitefish.
walking here,
I'm walking. I'm walking. I'm
Now
walking.
we're kind of cliche New Yorker stuff.
What?
That's not a New York that's existed for, I don't know, ever.
I know. The I'm walking here, New York. Is that what you're talking about?
I'm walking here in New York. Now people are like, oh, I'm sorry. Were you walking here? No, go ahead. This is like New York now. Like New York was so polite.
I
New York
don't know.
was-
I feel like I get like shoulder checked every time I go.
Actually that's because you don't get out of the way of anybody when you walk. I've seen how you walk.
Yeah,
like,
why should
"I'm
I get out of
walking
someone
here."
else's way? Why should I count out of other people's trajectory? That's not right.
Oh
I'm
yeah.
standing up for myself.
Them shoulder checking you. In fact, in a lot of instances, they were trying to get out of your way, and you're like, oh no, you don't. Ah.
That's not true.
No, it's not.
That's
You're
not
very
true.
polite.
Everyone's always like, "Are you from California?" Because like someone will bump into me, they'll shoulder check me, and I'll go, "Oh, I'm sorry."
I'm sorry.
They're like,
You're kind of like the
You're
crossing guy
from
like stepped on our
California.
foot, and I said, "I'm sorry." He was like, "You from California?" I'm like, "How can you tell?"
How do you know? You're apologizing. Who does that?
Apologize. Nobody does that.
Good.
Stop. Knock it off. It makes you a mark.
Good night, Fada.
"Yeah, let's go back to New York. That'd be fun."
New York would be totally fun. But first, we've got a show in Portland.
Yeah,
will-
which
Yeah.
I
be
swear,
awesome.
next time I'm gonna go to the Oregon Zoo.
Last
gonna
time
go
you're
to-
not gonna have time, huh?
I know I'm not gonna have time this time. Such
So-
a bummer. If I didn't have 500 teenagers coming to the zoo that Saturday, I would stay for the whole weekend. I would make it a thing. But I have to be back like midday Friday to finish the setup for this giant event on Saturday.
Giant. Yeah, that's a lot of teenagers, and it sounds like a big event. So
Yeah, and I'm like the point person, so it's, oh my god.
so it's on
Portland
you.
is-
Yeah.
Portland is one of my top 3 or 4 cities I've ever been to.
See, and I still feel like I haven't been
I know you've come
I
through.
spent like 2 months one day.
one day, 2 months, one day.
Or one time. I spent like 2 solid months in
Day
Portland.
felt
And
like 7 days, and the second day
I
felt like
did
2
not want to leave. I really did not want to leave. And maybe I don't maybe it wasn't the city, but but it was a nice backdrop at the very least. I'll give it that.
Nice.
And it's very easy to escape citydom in Portland, which is, it's a sort of Central Valley thing. It's not too difficult to escape to mountains or oceans. You can sort of get away from cityness pretty quickly. But Portland felt like you could almost just walk to hot springs or ocean or wooded things. Like you really, it was almost like the city itself was a premise of being a city, but would promise not to deliver citydom all the time. If that makes any sense to anybody at all. Okay. Is it time to say goodnight, Blair?
Is it? Wait, hold on. Is there anything else that I wanted to talk about for the show, week?
Noodles asking over and over in the chat room, Justin, what the thing behind you that's pink.
Yes, Noodle wants to know that. Yes.
He's
it's a secret.
What is it?
What, this?
It's a secret?
The blue and pink pattern thing.
Okay, so this is I don't know if I can show it. This is just a blanket. So this is actually in this. Well, I can move the camera. Watch this. So it's a whole bunch of paintings. And so to keep one painting from sticking face to face to another painting, you put blankets between them, and that's just a funky.
That's just a fun blanket. There you go,
it's
Noodles.
Just a funky blanket that's being utilized to keep paintings from. I had the window that's normally behind me that shows my place on the moon is in repair mode. And someday I'm gonna attempt again.
Are you having grommet issues?
There. Huh?
Grommet issues?
Grommet? It's because I haven't grommeted, because I haven't drilled the thing in, and I've been trying to do all sorts of stickum things, and then gave up, and then haven't circled back. But the funky coloring in the corner of the camera is just a blanket.
Just a blanket. There you go, Noodles. Mystery solved.
Yeah.
Kiki, did you have something else?
I've been trying to think. I know there was something else, but I forgot what it was.
You're almost done with calendars?
Yeah. I have fewer than 20 left.
Good.
Which is great. Oh,
I need to take it. That's good.
Yeah, I need to make t-shirts. I need to make new t-shirts. I'm out of some sizes. I need to make new ones. Should I talk to your dad about that again? Will he still have those old files?
Oh, yeah, he keeps all that stuff. Yeah. That should be easy.
All right.
Easy.
I gotta figure out if I want to put it on, I think, different t-shirt material. I gotta find different t-shirts.
Oh, yeah. Like a nicer, like a softer one, like an American Apparel style one.
Yeah, like a thin, soft cotton.
Yeah.
What is
they're just a couple more bucks a shirt usually, but
what is
he has access to all of those.
Heathered shirts? Is that a thing?
Heather is gray.
Is that a color, or is it a style of almost looks slightly distressed?
That's weathered.
I mean, that's a good description of it, but I feel like there's
Heather is a kind of gray. Heather gray.
See, this is why I have serious problems like ordering shirts and doing things like this, because there's all these details that you don't know until you dig into it. And then you're like, but I just want the shirt that's the nice shirt. And then they're like, well, you have to know whether it's a 4-ounce shirt or an 8-ounce shirt. What thickness do you want the t-shirt to be? And do you want it to be this boxy style, or do you want it to be cut this way? Or do you want that? Or do you want what? You need to know what Heather is. My friend is named Heather. That doesn't count.
So it's like if you want a women's cut, which is like curved, or if you want a box cut, which is probably what you want to do.
That's what I've done all along, is just a normal t-shirt.
Then what I think you're describing is a lighter weight premium cotton t-shirt.
Lighter, yes.
Is I think what it is, but he'll steer you in the right direction. It's literally his job to know.
Good.
But if you say, yeah, like I want like the nicer like American Apparel style t-shirts, he'll know exactly. Like he has
Okay.
a bunch of different manufacturers that he sources from. So
I think we should clarify
Cool.
too, just in full disclosure. Blair also works for American Apparel, which is why
yeah. No, I actually don't.
She was modeling for them.
Oh my God. No, I was not wearing a turtleneck leotard and nothing else on a white background.
What else could you wear when you've got a turtleneck leotard? Is that a thing? Has it ever been a thing?
That's an American Apparel thing. They love to put their models in like something with no pants. Like that's their thing.
But a leotard comes with a pants.
No, leotard is like the underpant bottom. Leotard has no legs. It looks like a swimsuit bottom.
Yeah, leotard. What people do gymnastics in, except for it's a leotard with a turtleneck.
Yeah,
turtleneck leotard.
What is it when it's like? Is it? I mean, I'm not talking yoga pants, but what's like dance pants then? Isn't that leotard also?
No, those are
leggings. I like calling them dance pants.
I think dance pants.
Excuse me, I need to go put on my dance pants now.
By the way, just in case anybody ever asks, I'm always wearing my dance pants.
I know you are.
God.
I mean, to be wearing them right now, we would never know. Here you go. There's an American Apparel turtleneck bodysuit. That's just what I was talking about.
Wow.
I love those. Like that's it's American Apparel loves to like. It's all right. It's after show, so I can throw a little shade, I think. But their ads are particularly specific. Let's say
they are.
If they're modeling a cardigan, then their model might only be wearing a cardigan.
Which is what makes their shirts nicer. That's what you're saying.
No, that's not what I'm saying at all. I'm saying that they are a stereotype for a very soft, kind of like luxurious t-shirt. That's like what one of the things they're known for is a very soft, thin.
Red cat's a thing when it comes to shirts.
It's more like a cotton percentage versus cotton poly versus like cotton.
How come thread count is such a big deal in sheets? But nobody talks about thread count anywhere else.
It's a lie.
It's all a lie.
It's a lie.
Thread count doesn't really matter. I've had 200 thread count sheets that feel like sandpaper, and 200 thread count sheets that feel like satin, and 200 thread count sheets that are just right in the middle.
Well, that's not really great that you're only comparing 200 thread count
No,
sheets.
that's what I'm saying. Like
up into the thousands, maybe it's just like amazing how many threads are touching your body at one time.
In liquid cotton.
Okay. Well, I think there's some 200 thread count sheets waiting for you. So say goodnight, Blair.
Kiki, did you remember what you wanted to talk about before we go? No.
Is that what she's doing?
No, I'm looking all over the place. Robot Kiki says good night.
Okay. Good night, Blair. Say good night, Justin.
Good night, Justin. Good night, Kiki.
Good night, everyone.
Now, why is this so?
I don't know what's going on anywhere. Good night, everyone. Thank you so much for joining us. Come back again next week. I'll be here. Well, not here. I'll be on Twitch on Friday at 1 p.m. Pacific time. Join me then. And I have my interview. This is
what? Santa hat? Periodic table. Chairs.
Interview nook.
Nook between two scientists. Or that's what Dave Friedel came up with. Or what was it?
Water bottle?
This Week in Science Conversations? I don't know, something like that. Okay. Get it back here.
So that would be Twisk.
There were some good ones people came up with. Yeah. Different names for conversations, but TWIS. So Blair, if we have time, come sit in my orange chairs with me.
Yeah, we can just chat.
That's right.
Just chat about animals for a little bit, or not animals.
whatever. We could just sit and talk in my chairs and talk about science and all sorts of things. It'll be amazing.
Yes. I love it.
And then Justin, if you want, you can come and sit in a chair and we can talk.
About animals or not about animals.
That would be a novel.
Exactly.
Yeah.
That's literally all the topics you can talk about. Animals or not animals.
Not animals.
These are the choices.
That's right. It's my studio. That is my corner for conversation. My interview nook. There is
Kiki's animal or not animal corner. with
Yeah,
Kiki.
there you go.
Hey, it's not an animal corner unless I put people and animals in it. Because people are animals. And plants are too.
Apparently.
God. That's what I learned today. Plants are animals too. Thanks for watching, everyone. Hope you can find something to eat out there.